yesterday I talked about how we make nanoparticle. Today I'll give you briefly about the survivin and what kind of work we are doing with the locked nucleic acid technology. So yesterday I introduced a little about what are the aptamers and what are the nucleic acid therapy and how you can make this nucleic acid therapy more or less vulnerable to the enzyme, nuclease enzyme, or the various other endonuclease enzymes, and how we make it more stable and useful for the cancer gene therapy. So here today I will talk about this survivin. We talk about this, the siRNA and how we make LNA nucleotides. So survivin, to introduce the survivin, yesterday I talked. Survivin is a protein, as name indicated. This helps cancer cell to survive. Not only cancer, all cells to survive. Especially with the cancer cell, all cancer, this is overexpressed. So overexpressed means it's starting from 5 times to 10 times more than the normal cell expression. And in cancer cell, this go from G0 to G1 case. So it's very aggressively means helping tumor cell to grow. As you know, the cancer cells, they are very active and metabolically they need high proteins or high metabolism due to high metabolism. So this is the protein which help the overexpression survival is help to the cell. So this survivin protein has been cloned in 1997, and after onward there are more than 5,000 papers on survival. So initially we thought survivin is only expressed in cancer cell, but later on we found the cancer, not only one cancer, it present in all cancers, and this protein is also present in chronic inflammation stages or the fibroblast or the cells which are remain in cell cycle arrest. They also have this survivin protein. So as I told you, it's first cloned in 1997. After that, we work on this protein. But our first paper came in 2001, where we use this antisense gene therapy, or we use a dominant active gene therapy. And here on, but there are a lot of publications come. We also continue to work on this survivin protein from the last 50 years. So this is the structure of the protein, and here this is the loop structure where it binds with the other survivin unit or apoptotic caspase or different pathways. So this is the cartoon here which shows the different stages of cell division. As I told you yesterday, this protein is very important. So you can imagine when there is a spindle formation occur during mitosis, this protein overexpressed. And if this protein is not there, there is no cell division. So this is very, very important to make it complex, and which we call as the chromosomal messenger complex, and where survivin plays a major role. So you can imagine if you target this protein or make a antisense or make a siRNA or some other form, you can downregulate the expression. If you downregulate the expression of this survivin, you can stop the growth of these spindle formation, and you can stop the mitosis cell division this way. So this is the cartoon here, how the cell signaling molecule are interfere with the survivin, and this as other signaling molecules, survivin also have a lot of the signaling common pathways which are responsible for cell division. Or if you stop this one, then you inhibit these caspases and these pathways, and you can induce the cell death. This paper we published 2010. This is the review article. So this is the paper which we published first time in JNCI. It's a very good journal. It's in clinical oncology. This is the first journal after review article of the Nature Medicine, where we use a dominant active protein. And I will give you some hints how we characterize, how we did this molecule and this gene, and how these proteins are interfering with the mitosis. So this is the paper first where we use the gene therapy, and we use the EL4 mouse lymphoma cell for developing the tumor. And you can see the difference between A and B. Here the tumor is small, here the tumor is large. We noticed that if the tumor is small, here the survivin expression less, the tumor aggression is less, and after some time tumor also come up. However, when the tumor is enlarged, when we use a siRNA to dominant active protein, or in combination with the immunogene therapy, where we use the CD86 or the CD80 or the B71 for the gene therapy, you can see here there is a complete regression regression in the tumor. And these tumors, they just come up again due to presence of this B7-1 immunogene therapy, so which act as a co-stimulation molecule. But later on, gene therapy has a lot of limitations, and if you notice, in 90s, the gene therapy impact factor go up, all general, but later on go down due to some regulatory actions of these gene therapy. So then we come up with an idea, so why not to make a protein therapy? Because proteins are less half-life and they remain in circulation for life. There is no question of integration with the DNA molecule or host chromosome. So we made a dominant active protein and we cargo this one with the polyarginine. People use that, we use R9, and it's enough to transfer this whole molecule of this dominant active survival protein within half an hour to any cell. Even chondrocyte, we use a chondrocyte for other study for the rheumatoid arthritis, and within half an hour, this molecule, whole molecule, get inside the cell through this the polyarginine molecule, which we tag on a terminal. So then we characterize this protein completely, how it goes, what are the size, and how this will interact with the different molecules in the cell, and how to get internalized in the cell. So as I told you yesterday, this helps for caspase activation and also mitochondrial depolarization, and which is very important. So this mitochondrial depolarization is responsible for activation of different caspases or responsible for metabolism of whole cell. So if this is depleted, so then there is no energy, and these cells induce cell death in these cancers. So later on, we published a review article, a lot of other, because there is a lot of correlation of survival protein with the BGF, with the heat shock proteins, and the hypoxia factor, which are responsible for tumor metastasis or growth of the tumor. So this is the cartoon here, how this, if you're interested, we published this one in Drug Discovery Today. In addition to survivin gene, there are splice variant of survivin as well. And as I told you, the survivin is conserved from so many years from nature, so still survivin is same molecule. But there are some splice variant inside the cell. And they are they have the regulatory roles for the activation or the action of this survivin protein. And these are various, and they are present in different stages and different cells differently. But this specifically, this survivin 3B, this is very, very important to play a role for regulation of survivin gene expression. So we published this data. If you're interested, you can see on this Drug Discovery Today. But here, in a cell division or in the cell growth or apoptosis, this has a major role on two factors, extrinsic factors and the intrinsic factors. So you can imagine if somebody inducing the cell death with the help of extrinsic factor from outside the cell through like phase, trail, or any other molecule, then this splice variant play a role. And this is the intrinsic factor. So in a nutshell, this splice variant of survivin, especially with the survivin 3B, have a major role for regulating the survivin expression. And this is a ping-pong mechanism. If survivin expression go up, then sometimes this variant form go down. So there is a quite regulatory role of this survivin splice variant. So from this, I mean, after 1997, there are a lot of people get attracted towards the survivin, how you target, and there are a lot of synthetic small molecule has been identified. And these are the molecules which are clinical phase trial. But unfortunately, all these molecules, they are toxic, and they are derived from either from the pesticide or the weedicide. And you can imagine, if you have a derivative from pesticide, you may end up with the side effect of these molecules. And these molecules, they're toxic. This is clinical phase trial, but this has been failed. This molecule, Abbott, is still in clinical phase trial. So this shows the importance of survivin inhibition. So if you have a molecule, small molecule, or have any regulatory which inhibits the survivin, this demonstrated that survivin will play a major role for tumor regression. But the importance of protein is, as I told you, is more appropriate because half-life is less and it has a less side effect, and after some time degraded and get excreted from the body. So this the protein we are playing with from the last 5-6 years, and we're making it dominant. And there are two forms of this dominant negative. We working on the 84 one, and there is another group they are working on 34 one. There is a difference between 34 and this one. This inhibit our dominant negative inhibit the BGF. But however, they don't inhibit the BGF expression. So we're making a dominant negative to double mutant one and playing around how we can use these protein for activation and the cell proliferation. So this is the paper we published recently in International Journal of Nanomedicine, where we make a nanoparticle of these survivin protein and use this for therapeutic application in a cancer cell. We tested this in colon cancer and the breast cancer, but similar mechanism we have found in prostate cancer as well. So if you see this one, I mean, interestingly, this survivin protein is present, into the cell within half an hour. And if you see here, this also downregulates the expression of stem cell molecules or other molecule which present in these stem cell. So interestingly, this survivin protein, which we have the dominant negative survivin protein bluish color, this is the dominant negative survivin protein which is present naturally inside the cell. So with the help of this one, this dimerase, and with this dimerization, this switch off this mechanism and reduce the cell death through mitochondria and mitochondrial depolarization or caspase activation, and this is the standard mechanism. So later on, we use the locked nucleic acid technology. Here we using locked nucleic acid and making a bispecific aptamer on the top of these nanoparticle. So the molecule which I'm introducing here, there are two molecules, nucleolin and the EpCAM. And these two molecules, they are also overexpressed in all tumor cells. And interestingly, these two molecules, nucleolin and EpCAM, they translocate from surface to the nucleus. So you can imagine if you design a nanoparticle which are tagged with these molecules on top of the nanoparticle, they'll trigger all the nanoparticle or the encapsulated protein, siRNA or any protein inside the cell. So this is the very interesting work which we did. Here, this is the nanoparticle, this is the chitosan nanoparticle. On top, we use alginate because we giving oral delivery system, so we want to protect our protein or these genes from low pH in the stomach. So here we use a sodium alginate. So these are the locked nucleic acid of EpCAM, which I says two protein very important. This is the nucleolin, and these proteins they're present on the surface of the cell. So if you design a nanoparticle with these locked nucleic acid technology, then they go inside the cell very rapidly. Within 30 minutes, these molecules go inside the cell. And if you see the stability of these two LNA locked nucleic acid, these EpCAM and the nucleolin, you can see here even after 24 hours they are still stable. So you can imagine if you have a aptamer or if you have a RNA based, and these molecules, these aptamers they are RNA based. So you can imagine RNA is very unstable. Within few minutes, within few seconds, RNA gets degraded. But if you use a locked nucleic acid technology, you can protect these one for a longer period of time, and you can save these aptamers. So this is the cartoon here, shows the mechanism, and I already showed you the detailed one. So now we demonstrated that these nanoparticle or these molecule, they are specific to the colon cancer cell, where we use the CACO2 cell. 98% get internalized, or 99 within few minutes go into the cell. However, in normal cell line, which we use for epithelial cell line, this is the FHS76. Here you haven't seen much interaction or internalization with these nano formulation. So this is the cartoon here. We have seen the expression and of these molecules, and here we use the standard Cifroid assay to see the mimicry, how these nano delivery system or these nanoparticles here we tagged with the bispecific aptamer log nucleic acid one, how they behave in different cancer cell lines. So these are the two cell lines which we tested here, and this is the normal cell line epithelial. So you can see here there is a significant internalization of these molecules. Here we did the loop assay. Here we take the intestine of these of mice and make a loop and see how they get these nanoparticles get internalized through the gut milieu. So this is the characterization as standard characterization. So we also did all characterization of these molecules. You can see here the efficacy of the loading capacity 15, and the associated efficacy is vary from 80 to 90%, which is quite good and significantly useful for our experiment. So this is the experiment which we did in the Cacto2 cell line with the xenograft model. Here you can see here we use the control or the untreated one. There is a growth of the tumor. When we use a nanobullet means a locked nucleic acid bispecific binding, a locked nucleic acid, these aptamer nucleoli. So you can see over there is a continuous regression of the tumor. And we tested it not only one cancer, we tested this one on different cancers. And here this is the apoptosis, how the apoptotic or the tunnel assay or the necrotic index goes. So here with the nanobullet, if you focus on this apoptotic index, is quite significantly high, and the tunnel assay is quite significantly. However, in control there is very less cell are inducing an apoptosis. So then we tested these nano formulation for other applications as well, and testing not only one cancer cell line, we tested these one on different cancer cell line, and as other people also demonstrated. So we also seen the stem cell, how the stem cell marker go with these nanoformulated treated mice, and how the PGP, because PGP is a molecule which is very, very important for multi-drug resistance. And here we demonstrated if you use these molecule in a combination, there is a regression or the sort significant inhibition in the growth in the PGP. So you can imagine in normal cell or the drug resistant cell, they have the high expression of PGP, and this is responsible for influx or outflux of these drugs. And with the help of this bispecific nanoparticle, you can skip this pathway because you don't need a receptor or you don't need a pump where your drug is pumping out. So that's another advantage is the nanotechnology. So we have seen that when we use these markers, the stem cell marker, these stem cell markers significantly down-regulating over there. And the mechanism go with the nucleolin pathway, and nucleolin is responsible for the down-regulation of stem cell marker as well. So then we see the wound healing assay, how this responsible for migration. So we tested all standard assay which everybody does. And then we tested another technology which is the DNA. So DNA is another form of locked nucleic acid form of this blocking. So we tested this one. You can see even after 14 days, this molecule with DNA is stable. So this is the beauty of these these locked nucleic acid technology. Here we have seen the marker of different stem cell. Here stem cell 133. Survivin, AppCam, you can see over here, they are all down-regulated with the help of this survivin siRNA. So then we tested these one on targeted and non-targeted nanoparticle. You can see over a targeted nanoparticle, they able to regress the tumor. If you see the survival curve of these experiment, there is a growth of the control mice, they die within few days. However, the other mice, they are not. So with the help of this targeted nanotechnology, where you target the tumors and these nanoparticles drag to the tumor site, more specifically with the help of these nucleolin and AppCam nanoparticle. So this shows that they have a very high survival rate. So this is the imaging system which we use with IVS, where we use the Sci-Fi file label with the nanoparticle or with our protein, and you can demonstrate that these go to the tumor site very specific. So then we did citroid assays, how these behaving, and we tested all these nanoparticles for angiogenic factor, because angiogenesis plays a major role for our tumor growth. So we tested all these markers. We have more than 45 different markers for angiogenesis. And as I told you, the survivin play a major role for cell survival, cell growth, and metastasis, and also responsible for the activation of VGF, hypoxia, and different these angiogenic factors. We tested all these one, and we tested these one on different cytokines, because the mice which we use, they are no doubt they are nude mice, but after 6 weeks, all the nude mice, they develop the immune system. So don't think means you are injecting tumor into the xenograft model of nude mice, you will not see the immune cell. No, it's not true. After 5-6 weeks, these nude mice, they start developing the immune system, and they have the role for regression of the tumor. And that's the reason sometimes if you inject the tumor into the mice after 5 weeks, you will not see the tumors. So sometimes tumor doesn't come up. So this is the reason, because these nude mice, they also start developing the immune system. So in our lab, we tested all these one in a routine way. So we tested more than 35 different apoprotic markers. You can see over here, these markers go up when we use these nanoparticles more specifically. However, in case of furoteuel, there's the less expression. In addition, we tested these one for different markers, AKT, all these yesterday you heard. So they are also impaired with this nanoparticle delivery system. So now we tested the pathology. All the pathology was normal, but blood analysis, when we did the blood analysis, there is a few issues with the blood analysis. When we use these survivin targeted nanoparticle, you can see here, there's not much significant difference in there. However, in case of void one, the WBC is going down. As I told you, these mice, the nude mice, after some time, they start developing the WBC, and they also have immune system and even cytokines present in, start expressing these nude mice. So we noticed that if you use these nanoparticles, they don't impair on these RBCs, and no effect on hemoglobin and other pathways. So, the nanoparticle, TAT nanoparticle which we use, they are quite safe as compared to the normal, and all the ranges are normal. So this is the mechanism here. The survivin protein, as I told you, this inhibit the PGP expression. They are responsible for inhibition of the stem cell marker CD33, which we tested. And here they have the pathway. And if you use the survivin as a dominant active, you can inhibit these pathway and induce these cell death in these mice. So this is the other paper this week came here. We're using lock nucleic acid technology, siRNA, by making a chimera of EpCAM, because EpCAM presents successively spontaneously on all tumors. So we exploit these EpCAM aptamer and make a chimera with the siRNA and use this technology for tumor in the retinoblastoma. So I'm not going into detail. I will show you the last mechanism here. So this is the interestingly, when we use the EpCAM aptamer lock nucleic acid and with the siRNA chimeras, so they interestingly bind with the EpCAM. And as I told you, EpCAM is a molecule which translocates from here into the nucleus. So if you have a nanoparticle, if you have attacked these aptamer with the help of these molecules, they go inside the nucleus. So that's the another advantage we found with the help of these lock nucleic acid technology. And interestingly, all these EpCAMs, these lock nucleic acid, they downregulate the expression of all these stem cell markers, which are very, very interesting and very much important for stem cell. As I told you yesterday, it's easier these days to treat the cancer cell, but it's very difficult to treat the cancer stem cell. So if you have a molecule or small molecule which inhibit the growth of these or the expression of these molecules, which is awesome. So these things are very important, and this paper has been published within 1 month in PLOS One. And later on, when it accepted, we thought we did a mistake. We should submit in higher impact factor journal. But anyway, this paper is published. If you want to read, this is recently. You put on Kanwar GR, and you can find all these publications. This is the another thing. Interestingly, we're doing in collaboration with the clinical people. They are using the AppCam, log nucleic acid technology, and using the drug for cancer therapy. Here, you can put any drug with these one. Only things you have to make a chimera. And it's very easy method. And if you have a drug and if you have CAM, this will cargo the whole drug from outside the cell to the nucleus. So you can imagine if your availability or the solubility of your drug have some problem, so you can use this molecule for making these happen. So we did all characterization of these molecules, and they are successfully going into the cell. So in nutshell, the message is, if you have a low nucleic acid technology or a low nucleic acid aptamer which expressed on surface or the molecule or surface of the cell, you can use the drugs and you can cargo whole molecule, protein, siRNA, aptamer, or drug from outside the cell, inside the cell, and within half an hour. All the assay which we tested all cell lines starting from brain cancer, liver, prostate, all cancer. Within half an hour, all cell take these chimeras inside the cell. And we tested in the chondrocyte and the other cell line, normal cell line. Within half an hour, they go inside the cell. Some normal cells don't take these so efficiently because the expression of these molecule or these molecules, they are not present in a normal cell. So which is again good thing. So you are targeting the cancer cell, and cancer cell, if you have a nanoparticle with tagged with these molecule, you can target the tumor more specifically and more targetedly. So this is the people get involved, and this means people here I have a collaboration with India, with the USA, and this is the funding where we all survive. Thank you very much.
So, one quick questions, if you have. Yeah.
So, you mentioned about tumor xenograft experiment in vivo. So, I wanted to know whether you injected intratumorally or it is by some other route.
Nanoparticle. No, some of these chimera we injected intratumorally. Yeah, but the most of these work which I did, we did with the target nanoparticle, we give oral route. Yeah, that's why it's, we tested systemically. What's the difference? Systemically go more, but with oral route you can easily tag these or target the tumor.
And second, you mentioned that in nude mice there is development of the immunity by the time. So will it be good to use in skid mice instead of nude mice?
Yeah, we tested the skid mice as well, but skid also have a lot of other problems. So in our previous experiment, we tested how much the role of NK cell and the other immune cell with the xenograft cell, where we deplete with the antibodies to NK cell in the bile. We haven't seen much in difference between these one. The reason is because when the tumor is big, then they also suppress some of these TGF-beta or other regulatory T regulations as well. So at that stage, we haven't seen much difference, but definitely you are absolutely right. When the tumors are big, with nude mice, they developed it. But skid mice also have some other issues with these things. So it's means demonstrator that immune cell in these ones, they don't have much role, but of course immune cell level. But there is we tested if we don't treat these the target nanoparticle, there is a 1 week or 2 week delay in the tumor growth. So that's there, yeah, obviously that's there. But when we deplete these with NK cell antibody or other immune cell, there is no change. So this shows that nanoparticle or target nanoparticle with this one in nude mice, they don't have much problem.